All right. I am recording. I have a few announcements. I made this announcement on Tuesday. Remember, your exam has been postponed until next Thursday. Okay, that's Thursday, February 20th. All right. Also, if you didn't hear this announcement, if you want to pick up a copy of problem set number 3, that's on cystic fibrosis, it is up here if you want to grab it. Also, I brought back your homework assignments for problem set number 2, 1 and 2, and if you didn't pick up problem set number 1, it's up here as well. I'll leave it up here. You can pick it up after class. All right. Any questions about the upcoming exam? All right. I do have one. Oh, question.
Did you mention that it was multiple choice?
It is. There's going to be 40 to 45 questions. I would say 80% of it, 85% of it is multiple choice. There are a few labeling. So like, for instance, I've already told the class this. Do you remember the unlabeled sodium, potassium, ATPase? It's those type of things, okay? So there's a few labeling. It's not short answer. There is no essay, okay? All right. Again, I think the best way to study for this exam is go through the lecture slides first. I'm not going to test you on anything we didn't talk about in class, okay? A big announcement here, I do have a formal review session. I have reserved the room. So it is going to be next Wednesday between 3 and 5 p.m. Wednesday, 3 to 5 p.m. in B45, Ruttan. All right, just right down the hall. Both sections are invited. Both sections, so there could be close to 180 students if everybody came. All right. Yes, question? If you can't make it, I always record it. Okay, I always record it. So you can catch up. If for some reason, as you know with this class, the audio goes out or something like that, I do have backup recordings. And it doesn't really change from semester to semester. So I can always, even before Wednesday, I can always load a previous review session. Okay? I would say if you could study as much as you can this weekend, bring questions, anything that you don't understand, I really appreciate the questions. Okay, then I can see where you're maybe a little fuzzy about certain concepts and we can go over it in detail. Okay, any other questions about the exam? All right, so what I'm going to do is I'm going to finish up with nervous system today, but I think what we're going to do is we're going to table the autonomic nervous system. If we have time next Tuesday, I will talk about the autonomic nervous system. If not, I'm going to table it until we get to the cardiovascular section for exam 2. We're going to need to know something about the autonomic nervous system, the sympathetic and parasympathetic, right before we talk about cardiac pacing, heart rate. Okay? As you know, sympathetic makes your heart race when you get anxious or when you get scared. Parasympathetic does the opposite. So it's nice to have that information right before we get into the electrophysiology of the heart and how the autonomic nervous system controls cardiac pacing. Okay? So we have a little leeway here. All right, so where I want to pick up then is, if you remember last time, let me go up to here. We did talk about Santiago Ramon Cajal. So here's where I want to pick up. First, I'm going to be talking about certain cell types within the cell. We'll start off by talking about Galil cells. All right? Galil cells are actually supportive cells. They support neurons. I don't know if you remember, last time we were actually talking about afferent and efferent peripheral neurons and what interneurons are, okay? Neurons are basically the cells that they are the ones that have the electrical signal. They signal other cells through dendrites in the central nervous system. These Galil cells are supportive cells, right? They actually support neurons. which is the nerve cells within your brain. So there's 4 types that I want you to know a little bit about. The first one are the astrocytes. Now, if you take a look at this slide right here, the astrocytes are in yellow right there. Oh, I'm sorry. The astrocytes are in green right here. These are the neurons in yellow. You can actually see the myelin sheath. Okay, so these are the neurons in yellow. The astrocytes are in green. You can actually see that the green cells, those astrocytes, they hold these cells into place, right? They actually are supportive cells. They hold the neurons very close to the blood supply. The other thing that astrocytes do, and I think this is one of the most important roles, is they have a high transport property. They're very active in ion transport. Okay, so they have a high transport activity. What they can do is they can modify the extracellular fluid that surrounds neurons. So they're able to keep the extracellular fluid that surrounds neurons within homeostatic ranges when it comes to electrolyte concentrations, pH, things like that. Okay, so those are astrocytes. All right, the next one here is the microglial cells. They're kind of these maroon-looking cells right there. All right, they're called microglial cells because they're actually really small at first. They are your immune cells within your brain. Maybe you didn't know that you actually had some immune cells within the brain. Once triggered or activated, these actually become macrophages. So if anyone's had some immunology, macrophages are phagocytic cells that can basically eat up foreign bodies within the brain. Okay, so these are your immune cells, the microglial cells. All right, so the next one I wanted to talk about is the ependymal cells, right? That's these brown cells that form a line right there. They are epithelial cells. They're basically the barrier between the cerebrospinal fluid and the interior of the brain that holds the neurons. What you'll notice is they're not like other epithelial cells in that they don't have a basement membrane. Those are other cells that kind of scaffolding that hold it in place. So they don't have a basement membrane, but they are ciliated, okay? So they play a role in developing, in secreting and creating the cerebrospinal fluid. And they're ciliated, so that when the cilia, these are kind of like projections within the cell. You can see it on this side and on the other side, actually. When they beat, like their hair-like structures, they keep the cerebrospinal fluid moving. Okay, so they're really important in producing cerebrospinal fluid and keeping it moving by the beating of the cilia. Ependymal cells, epithelial cells. Okay, and finally, this is a little tricky, okay? We're going to be talking about neurons today. We're going to be talking about what is a myelin sheath. So in the central nervous system, you can see these purple cells right in the center here. They wrap around the axon of neurons and create some kind of insulation. In the central nervous system, these are called oligodendrocytes. In the central nervous system, they're called oligodendrocytes. However, in the peripheral nervous system, we're going to be talking about motor neurons today. These cells that create the myelin sheath are called Schwann cells. So oligodendrocytes and Schwann cells do the same thing, right? They wrap around the axons of neurons, creating an insulation. They insulate the axon. That helps that it increases the conduction speed of the electrical signal. They also have some regenerative properties, repair properties. but it's not great. Remember, neurons are amitotic, so if they're completely severed, they don't divide and regenerate. So the oligodendrocytes and the Schwann cells have some repair capability. Okay? All right, any questions about Galil cells? Galil cells are supportive cells. All right? Most of the cells in your brain are actually Galil cells. All right, so here's some text to help you with it. Again, if you need something to read when you get home, it's exactly what I said. Astrocytes are supportive cells. They control the extracellular environment. Microglial cells are the immune system. When activated, they become macrophages, phagocytic cells that eat up foreign bodies. Ependymal cells are ciliated, and they're involved in the production of cerebrospinal fluid and movement. And then the oligodendrocytes are responsible for the myelin sheath. Pretty good? All right. Let's move from the central nervous system. Now, this is kind of a review from last Tuesday. Remember, the central nervous system it actually incorporates the brain and the spinal cord. That's what we're talking about. The peripheral nervous system are those neurons outside of the central nervous system and the spinal cord. So these are Galil cells of the peripheral nervous system. All right, the peripheral nervous system has satellite cells. Those are analogous, almost the same thing as astrocytes. And Schwann cells. Just like I said before, they're analogous to the oligodendrocytes. So this seems like really straightforward, but this is one of the questions that students miss the most on an exam, right? So central nervous system, oligodendrocytes. Peripheral nervous system, Schwann cells, okay? So the Schwann cells surround and form the myelin sheath, surround the larger nerve fibers, the axons, and they also play a role in regeneration and nerve signal conduction. As we'll learn when we talk about the length constant, right? You'll learn the length constant. It actually helps to increase conduction speed of that electrical signal down the axon when you have insulation. It's just like a copper wire. You know, electricians, they may put tape around a copper wire to create insulation. It actually helps with the speed of the electrical signal. Same thing here with the myelin sheath. All right, so let's zoom out, finish up the nervous system. Let's take a look at the brain as a whole. I'm just going to give you some very general description. Yes, question?
You had said that the satellite cells were like one of the central nervous?
Like the astrocytes, the supportive cells.
Thank you.
Yep. All right, any other questions? Okay, so let's zoom out. Let's take a look at the brain as a whole. For some individuals that are in the lab, the physiology lab, you've already had this information, so it's going to be review. Others who aren't in the lab, this may be new to you. Again, this is a very general overview, okay? All right, so let's actually talk about this forebrain first. The forebrain actually includes the cerebrum and the diencephalon here, okay? So let's concentrate on the forebrain, the cerebrum first. There are 4 different lobes that I want you to know a little bit about. The frontal lobe here in pink, the parietal lobe, the occipital lobe in the back of your head, it's green here, it's in green, and then the temporal lobe. All right, this is all part of the cerebrum. All right, the frontal lobe is more about higher thinking and reasoning, short-term memory, and dopamine-releasing neurons that are responsible for reward and attention. Okay, so that's the forebrain, the frontal lobe. Sure, absolutely. Okay, so the frontal lobe is really responsible for higher thinking and reasoning. It also is part of the primary motor cortex. I didn't say that before. It has plays a role in short-term memory. And it also has dopamine-releasing neurons that are responsible for reward and attention. Okay? Yeah, absolutely. All right, so the next one is the parietal lobe. The parietal lobe is really important in processing language and the somatosensory system. So what that means is, has anyone seen, maybe you have or not, the homunculin man when it comes to neuroscience, where he has a big face and big hands and big feet, right? What that is saying, this is part of the somatosensory. This is pain and temperature. You have more nerve per area in your face, your hands, and your feet, okay? So you're more sensitive to touch and pain in those areas. We do a 2-point discrimination test in the lab, and if you actually take a caliper and it has 2 prongs, you can distinguish between 2 points, say on your hand, the back of your hand, or close to your hand. But if you take the same distance and you put it on the back of your calf, because you don't have many neurons endings per area, it feels like only 1 prong, right? Even though it's 2, you just aren't, it's not as sensitive, right? Did everybody do that yesterday? Were there some people that did that yesterday in lab? No, okay. Maybe you're gonna do it tonight if you're in the Thursday night lab, okay? All right, so that's homunculin man, the somatosensory, right? That's all part of the parietal lobe, okay? The occipital lobe, this is pretty easy, is really just about visual processing. Visual processing. And then the temporal lobe is about language comprehension. The parietal lobe's about language processing. The temporal lobe is about language comprehension. Okay? It also, the temporal lobe is all about visual memories as well. So while the occipital lobe is visual processing, the temporal lobe, it plays a role in visual memories. Okay, pretty good? All right, so let's keep going here. Here, this pink area, can you see my arrow here? This pink area right here. When you do the brain dissection, this is very distinctive. This is called the corpus callosum, and it's basically the relay center between the right and left brain. Okay, that's pretty easy and very easy to see when you do a brain dissection. All right, then the cerebellum here in blue. The cerebellum is really important in control of movement and balance. Now, remember the frontal lobe also plays a role in primary motor cortex, right? But the cerebellum also plays a role in balance and posture, okay? Now let me just move to this next slide. How about this? This is actually a person. Here you can see with this image the cerebellum right here. It's a very distinctive structure within the brain. This person was born without a cerebellum. Isn't that crazy? It's just a cavity. Okay, so there's about 8 people in this world that live very normal lives just without a cerebellum. So they do have issues with, a little bit of issues with balance, but their forebrain frontal lobe actually takes over. So they have some vertigo, some kind of balance issues, but they can completely compensate with that frontal lobe. Okay? So no cerebellum, that's okay. It's okay. All right, so you can still live without that. All right, let's talk about these areas right here in the brain stem. So you have the midbrain and pons, which are basically the relay center between the cerebrum and the cerebellum and basically the spinal cord, okay? Specifically, the pons also plays a role in sleeping and dreaming. And then finally, I'm just going to talk about the medulla oblongata. Some people pronounce it medulla. It's okay, either way. Medulla oblongata. That's where some of the primary functions when it comes to heart rate and respiratory rate are. So you have the cardiovascular control center in the medulla oblongata, and that plays a role. It actually is tied very closely with the autonomic nervous system, the sympathetic and parasympathetic. It does play a role in cardiac pacing. And the respiratory control center is there as well. If you have changes in blood pH or changes in carbon dioxide levels, it can trigger your respiratory control center to either hyperventilate, increase your breathing rate, or hypoventilate, okay? Certain drugs can have effects on both of those centers as well. All right, so with that, here is how the peripheral nervous system is organized. Today we're going to, so a lot of people, I'm going to talk about neurons now, and a lot of people have asked me questions like, what are these neurons? Where do they come from? What role do they play? We are going to specifically be talking about the motor division today. Those are the somatic motor neurons, okay? And they play a role in muscle contraction, right? These neurons, this is voluntary. You trigger the activation of these motor neurons, and then you can move your muscles back and forth. Your muscles will contract. Yes?
I'm just interested in the cerebral.
Mm-hmm.
So, is there
yep.
any instance, like what instance would you have to get it removed?
Removed? Yeah, that's a good question. I don't know of any case where possibly there may be a tumor. That's the only time I would be able to think that that would be something you would want to remove. I'll look to see if there's someone that actually had a tumor that was associated with the cerebellum. Yes?
So you can see like cats who are wobbly because is it more common in like cats to not have one, or do they just have underdeveloped ones?
Okay, so with cats, what I understand is that they're not vaccinated and they actually are infected with a particular virus. Is it parvovirus?
I'm not sure.
Okay, so I think it's a virus. Do you know?
Well, not really specific, but I will say there's a lot of inner ear stuff with many animals that will affect their balance.
Sure, sure. I have a former student in this class who is in the PhD DVN program. I'm going to have her bring in both her cats because they have exactly what you're talking about, these balance issues. They were infected with the parvovirus, and it's exactly why. And I think it's a hypocerebellar response. Okay, so is that what you're kind of talking about?
I think so, yeah.
Yeah. So I should have Emily bring in her two cats. Yeah, it's actually pretty. I mean, they're terrific cats, and they do make it to the litter box, but boy, are they super off balance. But they but they really have a hypocerebellar response, I guess, yeah. So I'll have her explain it, actually. All right, and I'm sure you're right, too. I'm sure there's other, inner ear issues that could cause balance issues as well. Oh, yeah. Yeah, and my daughter has rabbits too, and once that happens too, a lot of times they don't make it. Yeah, rabbits.
It's very interesting how the ear can be so connected, especially when it becomes so important for function to the nervous system.
Yeah, absolutely. That's very true. All right, any other questions? Good discussion, very good discussion. Okay, so we're going to concentrate today on the motor division. But later, if we have time on Tuesday, we'll get to the autonomic nervous system, the sympathetic and parasympathetic. If we don't get to it Tuesday, we'll cover it right before the heart. Okay? All right, so let's go ahead and move on to neurons. Okay? I really like this, this lecture because it really brings a lot of things that we've already learned. It's almost a very easy lecture to give because you've learned, you have such a background now that I don't have to really build it up. I can just talk about it, okay? So here are some of the learning objectives. They're fairly easy for this particular section. Know the structure of a neuron, the anatomy. Understand the ionic basis of the action potential waveform. We already know about equilibrium potentials and conductance and driving force, so we're going to revisit that. Understand the effects of myelination, myelin sheath, on conduction speed, velocity, and we'll get to the neuromuscular junction right before we talk about muscle for exam 2. So it's kind of, you can see how everything builds on itself. All right. More learning objectives, but you can read those at home. These are more specific as to what we're going to be talking about today. All right, so here is the anatomy of the neuron. And like I always, kind of give you a heads up, this is one of them that is fair game for a labeling exercise. Okay, so you do need to know the anatomy of the neuron. Okay? So let's just start from the top here. You can see this is the cell body. It's also called the cell soma, S-O-M-A, soma. This is where the nucleus is, right? All of the DNA that's within the nucleus. The organelles like mitochondria here are in red. The endoplasmic reticulum here. Golgi apparatus, all of that is included within the cell soma. You know, if you've studied, I'm sure, even in high school, the cell, it has all the organelles that you would expect. It also has these long projections called dendrites. All right, dendrites. Dendrites, these long projections, actually receive signals from the axon terminal end of other neurons. Okay, so this is where the incoming signals relay their message. And then basically you get all of these signals called graded potentials within the cell soma. Okay, we'll talk more about that in just a second. Okay, just note that the bar on your left-hand side gives you some information about function as well. Okay, so you can see at the top, here's where the signal is received from the axon terminal end of other neurons. Remember we talked about presynaptic and postsynaptic? I'll start using those terms. This would be a postsynaptic neuron if we were receiving incoming signals from the presynaptic neurons. Okay, so signal reception is the first part here. So get this. This is pretty amazing. There's about 200,000 inputs, signal inputs from other neurons that can be signaling one neuron, okay? All of those signals, they sum in time and space. We're going to be talking about that in just a second. All that has to happen right here at the axon hillock is where all that signal is integrated. Okay, and if you can actually get a depolarization of only about 15 millivolts, remember resting membrane potential is about -70, you just have to reach -55 millivolts to trigger all of these voltage-gated sodium channels to get action potentials that propagate all along the axon. So this area right here with the myelin sheath wrapped around it. Now for these neurons, they're peripheral neurons. So these are Schwann cells, okay? So all of these Schwann cells wrap around the axon all the way down. They provide insulation. And the intermembrane nodal space, internodal space, is called the nodes of Ranvier. Okay? So internodal space is the nodes of Ranvier. The action potentials occur in the nodes of Ranvier. So it looks like it's hopping from internodal space to internodal space. It propagates all the way down the axon. This is called signal conduction. Signal conduction. And remember, the Schwann cells actually provide insulation that increase conduction speed. And then finally, it reaches the axon terminal end where neurotransmitters are released. This is called signal transmission. Okay? Everybody feel pretty good about these terms, the anatomy of a neuron? Pretty basic. It's going to get kind of complicated here in just a minute, so I want to make sure that everybody has the fundamentals. All right. So again, on the left-hand side is that bar. Here is the 4 functional zones. Make sure that you can label those as well. Signal reception. Incoming signal is received and converted to change in membrane voltage. Signal integration occurs at the axon hillock. All you need is a slight depolarization to activate all those voltage-gated sodium channels, which we talked about. Action potential travels down the axon. This is called signal conduction. And then at the axon terminal end where neurotransmitter is released, that's called signal transmission. All right, so I'm going to just bring it back to diffusion. I like to make all of these connections. If we had to rely on the neurotransmitters to actually diffuse down from the cell soma to the axon terminal end, if we were going to rely on diffusion alone, especially for giraffes that have a neuron that spans their entire neck region, could be a meter long, it would take 32 years. Okay, so that's not acceptable. What we have, animals have evolved, these cool little molecular motors. The ones that actually transport secretory vesicles or vesicles that house neurotransmitters, kinesins move from the cell soma down towards the axon terminal end, and dynein actually go in the opposite direction. They walk along microtubules. And the reason why I'm telling you this is because we're going to take a moment and actually I'm going to show you this video. This is what I was talking about before. This is called the inner life of the cell. Some of you may have seen this in general biology. But if you go about a minute in the inner life of the cell, this is an animation by Harvard. These are the microtubules. There's music to it too. They can polymerize and depolymerize. And then these are the kinesin molecules. Right? It uses ATP to actually literally walk along the microtubules. Right? It's pretty cool, actually. Dynein does the same thing, but it goes in the opposite direction. So, yeah, it's pretty incredible. So these are supposed to be vessels that are housing all of those neurotransmitters. So again, you have evolved these mechanisms to overcome the limitations of diffusion, which is pretty cool. I think that's really amazing. All right, so just wanted to roll that out. Okay, so here is where we begin with the exam. You will need to know the difference between what a graded potential is and what an action potential is. And what I would encourage you to do, I always like to help kind of organize the material so when you get home, it's easier to study for the exam. We're just going to put graded potentials. and we're going to list some characteristics, and we're going to compare that with action potentials. And you'll see how different they are, how similar but different. Okay, so I'm going to go into presentation mode now. I don't like going back and forth if I'm going to show a video out of presentation mode. Now we can go back into it. All right, so can you all see that in the back there? Okay, great. I have to put it a little bit lower because of the screen. Okay, so what you'll notice when you take a look at this is graded potentials, just observation right now. Graded potentials actually decay with distance. Let me find my arrow here somewhere. Anyway, my arrow's missing. Graded potentials are in green there. You can see with distance, the amplitude of that response diminishes, right? So I'm going to call that decay. It decays with distance, okay? Action potentials don't decay with distance. All right, so just some general observations. You can see graded potentials occur in the cell soma. Remember, that's the cell body. Action potentials occur along the axon. Just looking at this too, remember graded potentials decay with distance. But action potentials do not. Okay. All right, so let's start from the beginning though. Okay, so those were some general observations. Let's start from the graded potentials. Oh, here's my arrow, great. Okay, so along the dendrites and the cell body, how do we trigger these graded potentials? Usually it is a neurotransmitter that's released from the presynaptic neuron, right? Neurotransmitters released. It is a ligand-gated receptor that is an ion channel. Okay, ligand-gated receptor that is an ion channel. So this is the graded potential. Let's go ahead and put that on the board. Graded potentials are usually initiated by ligand-gated receptors. And I'm going to add another word there. Ligand-gated ionotropic receptors. Remember we talked about that on Tuesday. That means that the receptor is an ion channel. Receptor. With that, this signal could maybe open up a sodium channel. It could open up a different channel, like a chloride channel, right? So if sodium moves in, it's going to depolarize the cell. If chloride moves in, it's going to hyperpolarize the cell. You guys with me? Okay, so the signal could be depolarizing or hyperpolarizing. Okay. All right, so let's see here. If the signal is depolarizing, it actually moves the membrane voltage towards that threshold I was talking about, minus 55 millivolts. That means it's excitatory. If it moves it towards that threshold, it's actually more likely to fire an action potential at the axon hillock, right? So anytime these ligand-gated ionotropic receptors are activated and there's a depolarization event, moving it towards that minus 55 millivolts, it's excitatory. So you can see here, this is an excitatory potential. It's actually called an EPSP, excitatory postsynaptic potential. And it moves the membrane voltage from minus 70 to about minus 55, moving it closer to threshold. So this is a graded potential that moves the potential closer to threshold for firing and action potential. Pretty good? The typical ion would be sodium. Sodium moves into the cell, depolarizes it, moves it closer to threshold. EPSP. All right. Other ligand-gated ionotropic receptors may conduct chloride. Okay? Chloride would actually hyperpolarize the cell if it was moving into the cell. It would move it away from threshold. So these are called inhibitory postsynaptic potentials. IPSP, inhibitory postsynaptic potentials. It moves it farther from threshold for firing and action potential. All right, so now you can see with the graded potentials how they have to sum in time and space. You've got some that are depolarizing, some that are hyperpolarizing. You can see here in real neurons, it undergoes many, many EPSPs and IPSPs. Okay, so again, all of those signals have to integrate at the axon hillock. All right, so more building our chart here. We've already talked about these graded potentials can be excitatory or inhibitory. Also, they're kind of like it depends on how much neurotransmitter is released. They can be a weak signal or a strong signal, right? If you actually have a huge cluster of these ligand-gated receptors and a lot of neurotransmitter, it will cause a strong signal. And the stronger the stimulus, the greater the response. Or if there's just a few neurotransmitters that are released or a few receptors that are expressed, then you're going to get a weak response. And then finally, remember, it decays with distance at the bottom there. Oops, decays with distance. All right, so in this case, you can get a strong, you can have a strong or weak stimulus. And the response correlates with that, how strong or weak that stimulus is. All right, we're going to keep going here. Let's talk about temporal and spatial summation. All right, so all of these signals, these inputs, have to be summed in time and space. Let's take a look at temporal summation first. With temporal summation, just look at synapse 1. This is a presynaptic neuron, and this is a second presynaptic neuron, and then this is the postsynaptic neuron. So with temporal summation, all we're talking about is one neuron, just synapse 1, and its frequency of firing. Okay, frequency of firing. So let's take a look at this one. You can actually see this a little bit better, blowing up that chart. You can see if you stimulate the presynaptic neuron, it has a firing rate, you can depolarize the cell, and then it'll go back to its original value, right? If you increase the frequency, stimulate, you can actually reach summation. It will sum in time, right? You can get an even further depolarization if you increase the firing rate. Okay, temporal, just automatically think time. It's one synapse increasing its firing rate. Pretty good? All right, spatial summation on the other hand. Spatial summation is like this. If we have the cell soma here, all right, and here's the nucleus. And we have two presynaptic neurons here, like this. This is how they sum in space, okay? Think about it this way. Have you ever been like knee-deep in a lake that's completely placid, right? Meaning there's no ripples or anything like that. There's no waves. You take a stone, right, and you just drop it into the water. Right where that stone actually enters the water, the ripple is going to be the highest, right in the center, right? And then it's going to decay with distance, right? If you measure the amplitude of the wave that radiates out, it's going to decay with distance. That's how you should think about these graded potentials, right? So it's going to be the highest right where the neurotransmitter is released. And then it's going to decay with distance, as we talked about. So it decays with distance, it ripples out like this. And with this one, it's going to do the same thing, ripples out. Now, where they overlap is where they're going to sum in time, right? That's going to be sum in time. So what if this is releasing neurotransmitter that activates a sodium channel, and this one is activating a chloride channel? What's going to happen where the signal overlaps? It'll cancel each other out, right? So it will double if they're both depolarizing or excitatory. But if one is excitatory and one is inhibitory, they'll cancel each other out. Does that make sense to everyone? So summation occurs in time and in space with graded potentials. Okay, so we can actually say with graded potentials here, you can sum in time and space. It's going to be called temporal, that's time, and spatial summation. All right, pretty good. Again, this is just a slide this is from your textbook. It's kind of just giving you some information about all of the different signals. They sum in time and space. All that you need is the integration to occur in the axon hillock region. You just need a slight depolarization to reach threshold. What is threshold? It is the voltage required to open up all those voltage-gated sodium channels that are responsible for the action potentials to propagate down the axon. Okay, so taking a look at the action potentials, what initiates an action potential at the axon hillock is basically these are not ligand-gated ionotropic receptors. These are voltage-gated sodium channels. And we'll get to this in more detail after we finish up with the graded potentials. All right. Real neurons receive as many as 200,000 terminals. All right. So now I want to introduce a concept that's a little bit more complicated. Okay, we're going to talk about what the length constant is. All right, this is going to help you really understand a disease like multiple sclerosis. Okay, all right, so take a look at this. This is again telling you, again, we're back to graded potentials. These are ligand, see the pink triangle there? Ligand-gated ionotropic receptors. When the ligand binds to its receptor, it opens up a channel. In this case, this is a depolarization wave. Here at the bottom, this would be a weak stimulus. This one would be a stronger stimulus. And then again, it decays with distance. So what we're doing is we're measuring the amplitude of that wave. Just like I said, when you drop a stone into the water, you're measuring the height of the wave as it ripples out. Okay? Now, basically, you can characterize that decay with distance. This isn't in the time domain. Remember, this is in the spatial domain. Okay? And you can actually see that it is the distance over which change in the membrane potential will decrease by 63%. That seems awfully random, doesn't it? But at the end of the action potentials, I'm going to give you a formula. This is only going to be for extra credit. It won't be on your exam, but you can get extra credit, 5 points, if you do the length constant extra credit sheet, all right? Gives you some real insight on how to think about it. Okay, so what is lambda? Lambda is the length constant. It is basically characterizing the decay, okay? So it is the decay to 63%. It does fall out in the mathematics. Does seem random right now, but you can see that the pink line decays over a shorter distance. So here's where it decays 63%. And here is lambda for A. The lambda for A is a lot smaller than the lambda for B. Lambda for B, when it decays 63%, you can see it propagates farther. So the larger the lambda, the larger the length constant, the farther the signal propagates out, ripples out. Okay? Now, let's go back to the myelin sheath, right? Myelin sheath actually help to elevate lambda so that the signal propagates out farther. Those myelin sheath act as insulators, and it helps increase lambda. All right, so here's one equation. This is actually fair game for the exam, but it's only, you're not going to be running any numbers. It's just the relationships that you need to understand. Membrane resistance is the difficulty at which ions escape from the cell. If you have a myelin sheath and insulation, that membrane resistance is going to be high. Does that make sense? If you have insulation, if you have a myelin sheath, this membrane resistance is going to be high. So ions are not going to be able to escape, particularly potassium, and the signal, that electrical signal, is going to propagate farther down the axon. It actually correlates with conduction speed. Also, D is diameter. We're here at this particular equation down here. D is diameter. So the larger the diameter of the axon, the greater the lambda, the farther it propagates out. Why is it that the giant squid has a giant squid axon? Right? Because it has to run away from predators too. It has to run away. So basically, the larger the diameter, it doesn't have a myelin sheath like we do. So it has this huge diameter. It's a giant axon. And that actually helps with conduction speed and the propagation of that depolarization. Okay? And then the one underneath here, this 4 rho, it can also be thought of as for internal resistivity. Resistivity is talking about the internal intracellular fluid environment. It's like comparing a copper wire to rubber. Copper is really a great conductor. So these copper wires are going to have low resistivity and high conductivity, right? Rubber is not a good conductor, so it has high resistivity. All right, so this is in the denominator because it's going to have an inverse relationship with lambda. Here's how to think about it in cells. The higher the intracellular solute concentration, the higher the conductivity. So again, the squid axon, in our intracellular solute concentration is fairly low. Particularly for potassium, it's only like 5 to 15 millimolar. But for squid axon, it's like 400 millimolar. Again, you have to elevate that conductivity. So take a look at the difference between this top portion right here, unmyelinated axon, and this one down here, myelinated axon. Think about potassium in particular because you have a very high intracellular potassium concentration. Now I want you to think about them like billiard balls, okay? They're going to collide around, conk around, right? Sodium is going to enter in to the cell, for a ligand-gated receptor, and first thing it's going to do is collide with potassium because there's way more potassium inside the cell. So think about it like billiard balls. They collide with each other. They start to move down a dendrite or a cell soma or an axon. If the potassium can escape because there's no myelination, it won't propagate farther down the axon. It'll all escape out, and the signal will decay over a much shorter distance, right? But if there's myelin here, it doesn't allow the potassium to escape, so it propagates farther down the axon. Does that make sense? So that's kind of how to think about this. We'll have some practice. I know this is new for a lot of people, but this is how to think about the length constant, and we'll have some practice, some questions that I'll have you answer so that it'll be easier to think about for the exam. So if you need some text as to what I was just talking about, this is a really good one right here. And here is a table. We've already started making the table here, but if you want to have a reference, here's a table that's already made for you, okay? So differences between graded potentials and action potentials. I'm not going to go through it all because we've actually been already starting it. I'm going to start with action potential. You have like some brain teasers. We'll take a moment. We'll actually be able to get into groups and talk about this. Let me pull up this next one on action potentials. Try to take a look at this table when you get home. Bring questions on Tuesday as well. All right, so all right, it's thinking about it, getting it in presentation mode. Started already with the action potential. They don't decay with distance, and they're initiated by the voltage-gated sodium channels. Okay. All right. Here we go. There we go. Okay, so we've already talked about this table. Keep building it. Here's where we're at the axon hillock region. This is where all of those graded potentials, all of the input signals, they've all been summed in time and space. They come into the axon hillock region. This is where it all integrates. All you need is a 15 millivolt depolarization to reach threshold to activate these voltage-gated sodium channels. Okay, so you can see here at the top, here are some sub-threshold potentials. They don't quite make it to threshold. But then when they do, they fire action potentials. And you can see the amplitude of the action potentials are always the same. Why is that? Because it only occurs at the internodal spaces, the nodes of Ranvier. And you just have to reach threshold. There's the same amount of voltage-gated sodium channels at each node. That's why when they open to almost the exact same peak. All right? So they don't decay with distance. All right, so 4 action potentials each, the result of a stimulus strong enough to cause depolarization, are shown on the right. All right, so this is the big buildup. You've seen this diagram before. Remember? This is the ionic basis of the action potential waveform. This is where I really want you to put it all together. We've been alluding to it the whole time, but this is how you should think about the action potential waveform. First, the way that it's stimulated is a graded potential comes into the axon-hillock region, right? It has to be excitatory. It has to be a depolarization. It depolarizes the cell to threshold, and then you get those voltage-gated sodium channels to open. That's what this is right here. You get activation of the voltage-gated sodium channel. Take a look to see what it does to the membrane voltage. It pulls the membrane voltage closer to the equilibrium potential for sodium, right? You guys know that now. Why? Because the relative conductances are changing. If you use the chord conductance equation, you could calculate it out. You could calculate it out. Em equals GNA over G total times the equilibrium potential for sodium plus GK over G total times the equilibrium potential for potassium. You could calculate it out. At this point, I would say it's about 50-50. 50% of the conductance is due to sodium, 50% is due to potassium, okay? You get to the peak, right? That's about 90% is due to sodium, 10% is only due to potassium. Again, pulling it even closer to the equilibrium potential for sodium. And then those voltage-gated sodium channels inactivate. We've learned that already too, right? Then you get a second population of voltage-gated potassium channels that actually open right after the voltage-gated sodium channels inactivate. That changes the relative conductances, pulling the membrane voltage even closer to the equilibrium potential for potassium, right? Because now there's 2 populations. It's about 95% of the, 95% of the total conductance is due to potassium now. Only 5% is due to sodium because of the 2 populations of potassium channels. And then when that second population closes, then it goes back to rest. That's what's responsible for that after hyperpolarization. Okay, so that's should be review, right? If you hadn't learned all about the equilibrium potential and conductances, you wouldn't really appreciate this. But now you can actually see the ionic basis of the action potential waveform. Pretty good? Okay, so remember the voltage-gated sodium channel has the 3 different states, right? It has the open, it starts at closed, open, inactivated, right? Potassium, the voltage-gated potassium is just open, close. All right. If you need some people are more visual learners, some people really would rather see text associated with it. Here's a flow chart if you want to just look at the exact same process as a flow chart. Okay, pretty good? All right. So this is also part of that nerve simulation. We're going to do a few brain teasers here. All right. Now, like I said, I'm always giving you a heads up on what to actually focus on and study. I do want you to understand this graph. You do need to know how to read this graph. All right, so what we have here on the x-axis is time in milliseconds. Know these units too. Time in milliseconds. On the y-axis, membrane voltage in millivolts, okay? Now, what you have here is you have a typical action potential that's been elicited by, this is actually giant squid axon. This was done, it's using a technique called voltage clamping. In this case, it's open circuited, so you can actually see the change in membrane voltage, okay? Here's the black line, that's the change in membrane voltage with a typical action potential. The green line is the conductance for sodium. Remember, the sodium channel opens up first, so the conductance rises first, and then it inactivates and closes. Second, the delayed voltage-gated potassium channels then open, and then they close. So this G means conductance. The line going up means open, and this is a population effect. So just remember, this is a lot of different channels that open and then close for sodium. And then the potassium channels open, that second population, and close. Okay? You have the equilibrium potential right here for both. And then the two different scales for conductance in millisiemens per unit of area. All right, so some definitions, just review. Equilibrium potential. We're looking at the equilibrium potential for potassium. It's the membrane voltage required to keep potassium ions from moving down their concentration gradient. Equilibrium potential for sodium, right? It's the membrane voltage required to keep sodium ions from moving down their concentration gradient. Conductance. I would think of conductance like pathways. Remember, it's the ease at which ions move across the membrane. Threshold, by definition, this is the formal definition, membrane potential required to activate voltage-gated sodium channels. All right, here's a couple new ones. What is the overshoot? The overshoot is the difference between the peak of the action potential and 0 millivolts. Overshoot. Difference in membrane voltage between the action potential peak and 0 millivolts. And then this one, some people know this as the after hyperpolarization event. Some people have learned it by the undershoot. It is the difference in the membrane voltage between the most negative action potential value and the original resting membrane potential. All right, so let's play our little game here. In this computer simulation, you can vary things like sodium concentration outside, sodium concentration inside, potassium outside. You can add drugs like tetrodotoxin or pronase. We'll learn that in just a second. Or you can actually, let me go back here. You can vary temperature, okay? So here's what our action potential looks like first. Our temperature for the squid axon is 6.3 degrees Celsius. What if we increase the temperature to 25 degrees Celsius, room temperature? So I just changed it from 6 to 25. How would this affect the action potential waveform? I want you to think about this like enzymes. How does temperature affect enzymes? You can turn to your neighbor, take a guess. Who wants to take a guess? What's going to happen to the duration, the time of the action potential waveform? Anybody want to take a guess? Here's what it looked like before. I saw your hand go up. She was just scratching her head. What do you think? Yes. It would be faster, right? Enzymes, they actually progress faster over time in higher temperature. So what's going to happen? The voltage-gated sodium channel is going to open faster, inactivate faster, and close faster. Okay, so what's going to happen? You can see how the duration of the action potential waveform is really decreased. The time between activation, inactivation, and closing is much shorter, right? So here's where it was before with the dash line, and then in higher temperatures, the duration really decreases, right? That makes sense? All right. So let's try this one. Don't forget that the equilibrium potential does have temperature, so it's going to change slightly as well. All right, how about this one? Pronase is another drug that we can add. In this case, I've added 135 micrograms per mil of pronase. This is what it does. Take a look at the top. Pronase is a drug that clips the inactivation gate. It just completely cuts it off. What's going to happen to the action potential waveform if we do that? If it no longer has the ability to inactivate? You can talk to your neighbors again.
Well, actually, since inactivation comes from I just don't want to right because they just it doesn't have like the signal.
What do you think? What do you think? What's going to happen to the duration? Is it going to decrease or is it going to increase? Increase, right? Absolutely. So it actually prolongs the action potential, right? Because now it's in its open state and now you just are waiting for it to close, okay? I would argue too that the peak of the action potential is actually going to increase as well, right? All right, and that's kind of what happens here. You can actually see it prolongs the action potential waveform. All right, I'm going to do this one next time. Here's a good one. This is my last one, tetrodotoxin. Tetrodotoxin is a toxin that is isolated from pufferfish, right, fugu, right? What it does is it completely inhibits the voltage-gated sodium channel, all right? So it actually looks like this. Completely inhibits the voltage-gated sodium channel. So when you go to Japan and you ask for this type of sushi, they do make sushi out of tetrodotoxin. They actually have to, the chefs have to be, very, very well trained. They isolate it from ovaries and liver. If it is actually prepared correctly and you eat this sushi, it makes your tongue tingle. If it is not correct, if they don't actually prepare this correctly, it can inhibit your respiratory neurons and you will suffocate and die. Yes. So that's tetrodotoxin, right? All right. I'll leave you with that lovely note. I will see you next Tuesday. Have a wonderful weekend. If you didn't pick up your homework assignment, come on up now.
Hi. How'd it go?
Good. Yeah, let me go ahead and just... I know, it's so cold. Yes. Let me just shut this recording off. There we go.